Welcome back, Life Science learners, to our final segment for today. Let's look at a recap of what we've done today, and we get into our final segment looking at some application of what we've had to go through and look at how we understand how to unpack the concepts behind the structure of DNA. So we've spent some time looking at what are nucleic acids and where they are found. We've looked at, just recently in the last segment, we've looked at the discovery of DNA. We now know the structure of DNA, and if we were to explain that, you should be able to understand what the concepts of what a nucleotide is and how DNA is formed. So we've looked at essentially the structure of DNA. If we were to wrap that up, we would say it's a double helical structure. It's made up of two strands, and each strand is attached to each other in the middle by complementary base pairs, and that these complementary base pairs are made up of purine and pyrimidine bases. And we know that adenine, being a nitrogenous base pairs with thymine, and likewise cytosine with guanine. So we've looked at that structure. We've understood that it's a polynucleotide made up of these single small building blocks. Now let's use all of that and try and apply that to understanding and how that is tested in the context of different questions. So the first question, the table shows the percentage of composition of bases in DNA of cattle and an octopus. We've got to then use your knowledge of the structure of DNA to calculate the missing values of A and B in the table. Fill in the answers into the table. So I've got the table on the next page. I'm going to very quickly go to that table, and we can go back to the question. So here's a table that shows you the different organisms. So we've got a cattle and we've got octopus, and it essentially illustrates the percentage, and this is a percentage illustration of the composition of the different bases in DNA. And so here we're seeing that the table illustrates a 29% composition of adenine for the cattle, and it has also, for the octopus, it shows you a 33% composition of thymine. So I'd like to go back to the question, guys, so that we can read the question again and have a better understanding to that. So use your knowledge of the structure of DNA, and we've got to calculate the missing values of A and B in the table. Now guys, it's important that the question points us to using the structure of DNA to calculate. Now let's look at this. What's important for us is that we know that in this table that adenine has been given to us. So if we look at the cattle, the percentage of adenine is illustrated as 29%. If we look at the octopus, and that's given to us here, where we see that thymine is indicated as a percentage of 33%. Now guys, it's important that we pause at this point, and using our understanding of the complementary base pairs, let's try and link the rationale for solving this out. So if we've got DNA that's made up of complementary base pairs, and they've given to us a percentage of adenine, knowing that adenine and thymine are always proportionately equal, we can use that information to help us answer this question. So if DNA contains, for example, 20% adenine, it would mean that there would need to be 20% thymine. And that's purely from our understanding of the structure of DNA, that adenine and thymine are always complementary to each other. And this rule comes from Chargaff's rule, where he talks about when he extracted DNA, he found that no matter which species of organism it was, when he extracted DNA, he found that the composition of adenine and thymine were proportionately equal, and likewise the composition of cytosine and guanine were proportionately equal. Using that rule, we can now figure out the percentages of these missing bases. Let's try that. I hope you've got your calculator. You're going to be able to help me with that. So if we look at adenine, if adenine is 29% for cattle, it means that thymine also is 29%. And guys, if you can help me here, if we can add these up, so 29 plus 29 would give you the total percentage of what adenine plus thymine is. And so if you do your maths on this, so that equals to 58%. So 58% of the DNA is made up of adenine and thymine. Knowing that this is a percentage, it means that the balance of the percentage, so 100 minus 58, should give you what's left over. And so the leftover of bases, or what's remaining, is your cytosine and thymine. So 100 minus 58 is equal to 42. And if we try and unpack that, it means that the balance, which is guanine plus cytosine, equals to 42. This would mean that because guanine and cytosine are always equal, we would need to distribute that equally between the two. So we would divide that, and it would mean that each guanine and cytosine now contain or constitute 21% of that. So we've worked out that adenine makes up, and we said, let's look at that, 29%. So that would be, thymine would be 29%. And if we looked at guanine and cytosine, they would make up the balance of the 21%. And if we now were to apply the same rationale for looking at the octopus, we know that the octopus contains 33% of thymine, and we can quickly enter this on the table that there's a balance of 33% or proportionately equal amount that adenine is made up of. If we now add that up, we get 66%. So if we add 33 plus 33 is 66. And so it would mean that collectively these two, guanine and cytosine, have 100 subtract 66. 66. And we divide that by 2, and we can very quickly do that. So 100 minus 66 is 44. Am I right? 34, my bad. And if we divide that by 2, it gives us a percentage of 17% in each one of these. So it means that you've got to now use the rationale that adenine and thymine are equal proportionately, and that guanine and cytosine make up the proportion of the other bases. So collectively, A plus T plus G plus C collectively equal to 100, and use that to be able to work out the distribution proportionately between the complementary base pairs. So quite an easy question which often comes up, which you should be able to apply. Let's go into some terminology in terms of that we've just gone through in this segment. Give the correct biological terms for the following. And as I mentioned, the terms are crucial to understanding biology and life sciences. The base that pairs with thymine. Guys, this is easy. So we know that adenine and thymine always base pair together. So the base that pairs with thymine would be your adenine base pair. The next question, B. The building blocks of proteins. And guys, we've discussed that the building blocks of DNA are called nucleotides. And as we get into the next segments, it's important that we understand what are the building blocks of proteins as well. And so this is an extension question, and this refers to segments of amino acids that make up protein. So we know that amino acids put together form proteins, or the monomers of proteins are called amino acids. Let's look at C. The bonds that hold the two polynucleotide strands of DNA molecules together. So we said that DNA is double-stranded and it has these complementary base pairs. And they are held together in the middle by what we refer to as your hydrogen bonds. So we've got your hydrogen bonds that hold these complementary base pairs together. And so it's our hydrogen bonds that hold them together. D. Repeating units or monomers that form a nucleic acid. So the repeating monomers of nucleic acids are called nucleotides. So the DNA is made up of nucleotides, and we know that we've got RNA nucleotides and DNA nucleotides that produce different types of nucleic acids. Let's look at E. Sugar that forms part of DNA. And in our image earlier on, I referred to that pentose sugar, and that pentose sugar that forms DNA is called a deoxyribose sugar. So it's a deoxyribose sugar. The shape of DNA molecule is the next question, and as I mentioned, that DNA is a double helical structure. So DNA is a helical structure, or in some cases you can refer to that as being a helix. So it's helical, forms a helix. Let's move on to the next question, folks. The figure represents a part of a nucleic acid molecule. Study the diagram and answer the questions that follow. So let's move on to the figure. So here we're seeing a figure that shows you part of a DNA molecule. Let's try and study this figure and answer the questions that follow. Again, this is a very schematic representation of DNA, and you can see that it has two strands, and I'm not going to scribble on this yet. So you've got the one strand here, and you have the other strand, which is on the other end. We've got some structures that are labeled, and these clearly indicate a specific shape, as you would see, and these shapes are unique in that you can see that only this structure number 4 can connect with number 3, and so that uniqueness is based on the complementary base pairing. Important to this diagram is a key that is given, and this key indicates that adenine is represented using the letter A and cytosine using the letter C. So in this diagram, we can see that adenine is here, and it's identified with this specific shape here, which I'm pointing to now. We know that cytosine has been labeled, and cytosine has been identified with that unique shape. So using our principles of complementary base pair, we know that adenine will always base pair with a thymine, so this complementary nitrogenous base on the other end is going to be a thymine, and likewise cytosine base pairs with guanine, and hence that number 5 is going to be a guanine base. So guys, using the shape of A, we can use that to help us figure out what the base for number 4 and 3 are. So based on the shape, knowing that adenine has a unique arrow-pointed shape, we can use that to draw inference that number 4 would also be an adenine. And likewise, based on that, we know that number 6, which we identified as thymine, will be also indicative of what number 3 would be. So based on that, we've now used that information to figure out that number 4 would be adenine and number 3 would be thymine. And so guys, as we wrap this section up, it's important that we try and link back to where we started. Identify the nucleic acid shown in the figure, and we know that this nucleic acid, again, is DNA, and the reasons would be that, firstly, DNA is a double-stranded structure. We can see that it contains 2 strands. And more importantly, we've just identified that it contains a thymine base. And that's unique to only DNA. And DNA contains thymine. And using that, we're able to identify and link our understanding of the uniqueness of DNA is that it's double-stranded and it contains a thymine nitrogenous base. Guys, you've done well. We've gone through this segment looking at the application of the structure of DNA. In our lesson today, we've looked at DNA as a molecule that codes for life. We've tried to unpack what the structure of DNA is, and we've tried to understand how we can use that information, especially about complementary base pairing, to work out the composition of nucleotides in a DNA molecule. So guys, you've done well. Go well, keep safe, and looking forward to seeing you soon.